Hi everyone, my name is Manuel Copas and I'm going to be with you for a couple of hours this afternoon, Friday afternoon. Hopefully we'll make it fun enough not to make you sleep. Maybe I can introduce myself. So I'm a data scientist by training. I did most of my training at the University of Cambridge, and now I'm senior lecturer in genomics. So I'm going to give you two sort of sections today. So I'm going to talk first, give you some background on the, I guess, theoretical framework to then go into the second part of this session, which will be practical. So for this presentation, I will basically give you some ideas about how to analyze and interpret from a bioinformatic point of view. Hi. The whole genomes. And then after that, I will require you guys to do some data analysis using Excel. So this presentation, I'm going to record it, so it'll be available in Panopto. So it's being recorded right now in Panopto. But the actual practical is not going to be recorded because it's more like hands-on. Are you able to see this, or shall I turn off the lights a little bit so you can see better? Is that better? I take that that's better. So I'm going to talk about concepts around something called precision medicine, which is basically, to a great extent, is the usage of genetic data in order for us to help diagnose and treat patients. And this idea that not one size fits all. How do we make sure of the specifics that each person has with regards of medication? We all know that you give the same drug to different patients. Some patients may have adverse drug reactions. So, and a lot of that is explained not just by the environment, but also by the genetics. I'm going to give you a very, very sort of brief presentation of how currently in the clinic the genetic data is used to help diagnose patients, and how we interpret and classify the bits of DNA that are potentially pathogenic. I'm also going to give you examples of diseases that people die of. In specific, I'm going to be talking about the genetics of Alzheimer's disease. And then I'm going to be talking a little bit about some of the bioinformatics databases that we use to help interpret genetic data in patients. So for the purposes of this session, personalized medicine and precision medicine is going to be the same. And this idea revolves around wanting to have much more specific targeted diagnostics and treatments, rather than every patient that's got Alzheimer's disease, they're going to have the same treatment, the same diagnosis. Now we want to go really into the specific characteristics that each patient has. The idea that we can give each patient the right drug at the right time with the right dose, and then this idea of P4 medicine, participatory medicine, preventative, personalized, and predictive. And it's very much data-driven, genetic data and symptoms data and historical clinical reports, all data about the patient. So how can we leverage this data to help patients have better health care? So what is genomics? So genomics is for the genome what genes are for genetics. So before we had the Human Genome Project, that is the map of the whole length of all of the genetics for each species, in this case the human being species, we only had genes. So genetics dealt with genes in isolation. Genomics deals with all of the genes. So genomics means all of the genes. And so genomics studies the structure, function, and evolution and mapping of genomes. As I said, the genome is an organism's complete set of DNA, including all of its genes, but also the mitochondrial genome, for instance. And genetics refers to the study of individual genes. So the Human Genome Project was, in my opinion, one of the most important projects in the 20th century, turn of the 20th century to the 21st century. And the goal was to determine the sequence of nucleotides, as we call them, or base pairs that make up the human DNA, so that we can then identify where the physical location of each of the genes in the patient is. And then in the same way that we have Google Maps, where in order to go from point A to point B, you need to know the coordinates. So in genomics, in order to be able to know where the genes are, where the regulatory regions are, where the introns are, where the exons are, we need to have the specific coordinates. And so by having the coordinates, then we can better understand differences in the DNA and how those differences actually are affecting the health of the organism. So the Human Genome Project, the finished human genome, is a mosaic. So it doesn't really represent one individual, but it's a mosaic of individuals. And it's called a reference genome. So the reference genome is, as I say, a consensus sequence of DNA that help us then compare whether any other individual has any difference between the reference and the individual itself. And so we don't talk about mutations in genomics, we talk about variants. So a variant is a difference that we have with respect to the reference genome. And so genomics is the study of the variants and its potential effects with respect to the reference genome. So the genome of any given individual is unique, and it help us understand diseases where the presence of specific changes can help us understand whether there is a deleterious effect. For instance, whether any specific variants can increase the risk of developing cancer, as well as what the right type of medication would be for a specific patient based on the enzymes that metabolize drugs, which are also encoded in the genome. So genetic variation, as I said earlier, is how one individual differs from a reference genome. So if we have here a reference genome for a specific random region, and we see that the colored set of nucleotides or base pairs are different between an individual and the reference. So the simplest type of variation that we can have is what we call a SNP. So a SNP would be, let's say, that we change that T into a C. So we change one letter out of the 3 billion letters that we have. We change one letter for another. That's a SNP, a single nucleotide polymorphism. It's the simplest type of variant that we can have. And SNPs are the workhorse for genomics analysis. Okay? The vast majority of the variants that we find, and each individual may have millions of variants. In fact, on average, each individual has around 5 million SNP variants only. There are other type of variants than SNPs. We have other like copy number variants or insertions, deletions, but just SNPs, every individual has on average around between 4 to 5 million. And the vast majority don't do anything, but there are a few that can have a tremendous effect in the health of the individual. And so we want to understand how those different SNPs are contributing to disease risk, to health patterns, and so on. So single nucleotide polymorphisms. So they are the commonest of variants, and each SNP represents a difference in a single DNA building block called a nucleotide. There are roughly between 4 to 5 million SNPs in a person's genome, and some SNPs may help predict an individual's response to certain drugs, environmental factors, and risk of developing diseases. The diseases that people die of, type 2 diabetes, cardiovascular disease, mental, autoimmune diseases. And SNPs can be used to track the inheritance of those diseases across generations, helping us have a better understanding of the genetic contributions to disease risk. So here we have an example of one SNP. I'm going to make this a bit bigger. I'm not sure if we can see it very clearly. So we have a SNP here which is called RS42938. So that's the ID, the identification of the SNP. And here we can see the coordinates. So this SNP is in chromosome 19, position 44,908,684. Right? So it means that within that chromosome, at that position, the reference is a T and it has been changed to a C. So that change from T to a C at that position in that chromosome is what we call this SNP. And so we also see that actually this SNP has a consequence. It's within one gene called ApoE, and as it happens, if you have that change from T to C, it causes a missense variant. A missense variant means that the protein sequence of that gene at that position is changed by an amino acid. Because you know that DNA is transcribed to RNA, RNA is then translated into sequence of amino acids, and that sequence of amino acid is what then conditions the structure of the protein, and the structure of the protein gives the protein its function. And as we know, proteins carry out almost an infinity of functions, from being enzymes to be structural scaffold of the cell, and so on. So basically, you have that little change at that position, you're going to change an amino acid of the protein resulting from ApoE, which is a gene in the organism. Is it clear? Any questions? Okay. So the issue is that we may find SNPs, but then we need to understand what those SNPs function, or what the effect of those SNPs is. So the type of questions a clinician would want to ask himself or herself is, does this sequence change in the context of genetic background provide an explanation for a specific phenotype? Or is an individual who inherits this sequence change likely to develop that particular phenotype? So the American College for Medical Genetics has suggested a system for classifying any given variant that we observed in the genome of an individual. And so from more pathogenic to least pathogenic, there are five categories. So it can be pathogenic, that change can be pathogenic, it can be likely pathogenic, it can be uncertain significance, it can be likely benign or benign. So basically, every single variant that you can see, you're going to want to identify those that are pathogenic or likely pathogenic. The other three, we're not that interested. Okay, so every variant we classify according to this standard, which now has become adopted worldwide. But how are these variant data produced? There are different technologies with which we can generate genomic data from a patient. So we can have array data, which basically consists on 1 million SNPs that are tested at specific positions, and then we allow, you have to know in advance those SNPs, where they are, where they are located, and then it allows us to be able to identify, it allows us to do genotyping. Have you heard of that word, genotyping? So arrays are okay for genotyping. Or you can also perform Sanger sequencing, which allows us to sequence, show fragments of DNA at any given position. Or we can do sequencing. So there are two main types of sequencing. And sequencing means that you actually look at every single position across a particular region or the whole genome. So exon sequencing means you are identifying variants across regions within protein coding positions in the gene, in the genome. So protein coding regions are the regions that encode for proteins in the genes. So you have introns and exons. Genes have bits that are translated into protein and bits that are not translated into protein. So protein coding regions account for about 2% of the whole length of the whole genome, but they are around 80% of disease-causing mutations. So they are very important because these are the bits that get translated into proteins. But of course, you're going to miss a lot of interesting data, potentially, and variants outside of the coding regions. The good thing about exome sequencing is that it costs less and it's not as heavy as whole genome sequencing. Whole genome sequencing would take as much as 200 gigs of data. So it's it's very heavy, very difficult. But the good thing is apart from that, it's also quite expensive. But the good thing about it is that you're going to span the whole length of the genome, meaning that you'll be able to have all of the potential variation that you would be interested in. So this is how whole genome sequencing works. So have you been explained what next generation sequencing is? Have you come across NGS? Would you like me to remind you a little bit what NGS is? Now, NGS is basically these massive machines from a company, for instance, Illumina, whereby you cut your DNA sample into millions of fragments. Then those fragments, you feed them in the machine, and then the machine reads in parallel those millions of fragments. And those millions of fragments allow you to, they get digitalized. So from being chemical compounds, you get a massive file, which you then, with a massive supercomputer, you have to, like a puzzle, reconstruct its exact order based on the reference genome. It's like a massive puzzle that you have to redo based on the GT2 fragments that you have across the length of the whole genome. So here, if we had a hypothetical chromosome, here you have short reads, so the short fragments that get stacked, because there's a lot of redundancy. So you get all of these short fragments aligned according to the specific sequence, matching the reference sequence of our reference chromosome. And so the idea is that by stacking all of these fragments, if we find, I don't know what's happening here, if we find a position which is different from the reference by consensus of many fragments that overlap each other, we have sufficient confidence that actually here there is a variant. You probably can see it. So we have gray out, so each position corresponds to a nucleotide of the reference sequence. And here what we are looking is at whether there are any specific differences between what we have in the sequence of the fragments that have been correctly aligned against the reference sequence. And so if we find any sequence, this software is going to color the positions where you have different sequence from the reference. And it is precisely these variants, these differences, what we're interested in interpreting, to see whether they're actually causing, for instance, the change of an amino acid in a protein. Any questions? Any thoughts? Am I talking too fast? So as I say, the whole of genomics is not to look at the things that are the same as the reference, which here we have grayed out, but we look at the things that are different from the reference. The consensus reference human genome, which here we have colored. And by looking at these specific positions, which are different between the individual that you are sequencing or genotyping, then we can begin to classify them to see whether they have a potential pathogenic effect or not. That's really the key with genomics. So next generation sequencing, which I have tried to explain here more or less how it works, is the most comprehensive type of variant data production that is available. and it uses massively parallel sequencing platforms to understand the positions of nucleotides in the DNA sample that are different. So, just to reiterate the differences, it's important to understand that the two main types of sequencing that we have in humans, the exome, which only sequences coding regions, is 50% cheaper than the whole genome. It's only around 2 to 4 gigs of volume of data, and the diagnostic rate or yield is around 30%. Whereas the whole genome, it costs around $1,000. It occupies somewhere around 200 gigs. It has much wider and uniform coverage of the genome, and it provides answer for a lot more interpretation. So what this means is that exome is going to miss potentially, if you just look at the coding regions variance to explain a patient that has a congenital, that is a disease they were born with, just looking at the exome you're going to miss a lot more than if you look at the whole genome. That's kind of obvious. So what's the difference between genotype and phenotype? So genotype is really what we see at the level of the DNA, and phenotype is what we see at the level of the person or organism. So here the phenotype is trisomy 21 or Down syndrome, but you can have many different types of symptoms or phenotypes. Learning disability could be a phenotype, characteristic facial features, congenital heart disease, visual impairment. So all of these are things that we observe at the level of the individual, and that's what we call phenotype. Whereas what we see at the level of the sequence is what we call genotype. Another interesting concept is dominant heredity versus recessive, and this makes sense in the context of Mendel's laws. So you might have come across the Mendel's laws. He was this sort of first person that looked at, with peas, how inheritance worked. And the reason he was able to find the genetic patterns or the inheritance patterns was because he was very lucky and I guess wise to be able to focus on traits that are monogenic. So in peas, the color of the pea is produced by one gene, right? And the color of the flower of peas is also conditioned by one gene. So what I want to say here is that Mendel's laws work in the context of phenotypes that are monogenic, that are caused by one gene. So what that means, for instance, red hair color, right? So I'm a carrier of red hair, but because I also carry black hair, and black hair is dominant, I only need one copy of the two chromosomal copies I have, one for my mom, one for my dad. We all have two pairs of chromosomes, right? So we have two pairs of genes. So if for the same gene I have two different versions, one version is red hair and one version is black hair, the dominant character or the dominant phenotype is the one that only needs one of the two copies that we've inherited for it to be expressed. Okay? Whereas recessive, in this case with the hair color, red hair, is a recessive trait because you need the the hair gene, to have both copies for it to be expressed. But this dominant recessive paradigm doesn't work for most diseases that people are affected in health, because most diseases are polygenic. Yeah, so diabetes is highly polygenic. Cancer is highly polygenic. You know, the main diseases people die of in the West, they are highly polygenic. That means that many genes contribute in a small way, yet meaningful, to that, to developing the disease or not. And when we are talking about polygenic diseases or traits, the laws of Mendel's don't work, because they only work when we have monogenic traits. Okay? So here I've made the distinction between Mendelian disease and complex diseases. So everything that, or all of the diseases that involve more than one gene, we call them complex diseases or polygenic. So Mendelian diseases are rare, whereas complex diseases are common. Most people die of those. Mendelian disease are highly penetrant. Penetrance means that the probability that by having that genetic change you're going to develop the disease. So the most penetrant, one of the most penetrant variants that we know is the BRCA gene variants, specific BRCA gene. So BRCA is a well-known breast cancer gene. If you have a specific mutation in that gene, it's highly penetrant, meaning that most people that have that variant, they're likely to develop, but not all of them develop the disease. Maybe the most penetrant of all the variants could be around 80%. So 80% of women that have that variant are going to develop breast cancer, but not all of them. So for that reason, that's why we call penetrance, because you may have variants that are highly pathogenic but not very penetrant, meaning that even though they are pathogenic, that they don't express the phenotype very often. Okay? So the Mendelian diseases tend to be highly penetrant, whereas complex diseases tend to have variable penetrance. The Mendelian diseases follow the laws of Mendel, whereas the complex diseases have unclear and variable models of inheritance. Mendelian diseases have minimal to mild environmental influences, meaning if you have that variant, it doesn't matter whether you do exercise or you don't do exercise, you're most likely going to develop. Whereas polygenic diseases do have a much greater contribution from the environment, meaning that, you know, if you have a, let's say, melanoma, which is in great part conditioned by ultraviolet exposition. So if you don't go to the beach often, then most likely you won't develop melanoma. Okay? Or, you know, cardiovascular disease. If you do exercise, take care of your diet, don't smoke, don't drink, you're going to have lower chances of developing cardiovascular disease. So polygenic diseases have much more influence from environmental factors than Mendelian diseases. And Mendelian diseases often produce gross perturbations in protein function, whereas complex diseases tend to have minor common variations that are very, very small contributions. You can have millions of contributions from different variants across 10,000 genes for diabetes, for instance. Examples of Mendelian diseases: sickle cell anemia, cystic fibrosis and Duchenne muscular dystrophy, whereas complex diseases examples include cardiovascular disease, diabetes, cancers, and psychiatric disorders. Any questions so far? So we have two complex diseases here at two opposite ends of the environmental spectrum in terms of influence. So type 1 diabetes is highly polymorphic, highly polygenic, but the genetic risk is much greater than the contribution to developing the disease from environmental factors. So basically, that's why type 1 diabetes tend to be in children. So you don't really, no matter what you eat, you're most likely going to develop it. Whereas type 2 diabetes is the opposite. So if you have bad diets, it's a lot more influenced by the environment. So for type 2 diabetes, the environmental contribution would be a lot bigger than the genetics. Here we have the opposite. So melanoma, which is a type of skin cancer, if you have high genetic risk, it doesn't matter if you don't get exposure to the sun. So for melanoma, you have the most contributing factor is the non-genetic environmental risks. The good thing about genetics and genomics is that these variants that we are now able to identify, because we have the map of the human genome, we can then estimate a priori the likelihood of a patient to developing a disease, just based on looking at their genome. And so, although I'm not claiming that the genome data is determinant, it is certainly contributing. And so for the first time we can really look at the genome of a person and have a very strong marker for risk prediction without even knowing or understanding their clinical history, right? So the complex diseases, so all complex diseases, as I said, have many contributions from different genes. And the way for us to really understand the genetic contributions for complex diseases is through a technology which we call genome-wide association studies, or GWAS. So GWAS uses SNPs to help understand what genetic variants are more likely to develop the disease. And based on those genetic variants, then we can stratify high-risk individuals. So patients that have greater number of risk variants are more likely to develop the disease. So the way genome-wide association studies work is really a statistical artifact whereby I don't think, I thought I had a figure there. So I have to explain, but basically the way genome-wide association studies work, you have controls, you have patients. If you find specific variants that are more frequent in patients versus controls, then those variants are associated to the development of the phenotype, of the risk. If we add weights, because not all variants contribute the same amount, we can actually add weights to each of the variants that need to be present that contribute to the disease. Then we can develop something called polygenic risk scores. So polygenic risk scores allow us to sum all of the weights of the different allele contributions, of the different variant contributions that we observed in a patient, and then it allows us to see whether a patient is high risk or low risk by summing, as I said, the presence of all weighted variants that we observe in a patient. And they're very useful to help stratify high-risk individuals. So here we have an example of polygenic risk that basically uses GWAS variants, and then it provides a weight for each of the variants. So you may remember I mentioned earlier that SNP in RS429358, which was part of the ApoE. So I said that SNP is a missense mutation, and so it means it changes one amino acid for the gene ApoE. And as it happens, and I didn't tell you this because I was hoping to sort of make a little punch now, if you have that little change, your risk of developing Alzheimer's disease increases by 253%. I'm going to say that again. If you have that for 3 billion letters, you have that T changes into a C at that position, and your risk of developing Alzheimer's disease increases by 253%. It's not bad, is it? So if you then sum the weights of all of the SNPs that have been identified contributing to Alzheimer's disease, then you're going to get a distribution of patients whereby those that are in the 90th percentile, say, or the 95th percentile, are going to be 3 or 5 times greater average risk than the average person. Okay, so that's how it works. So genetics is never going to say you are going to have it or you are not going to have it. What it's going to say is you have X times probability to develop it compared to average. And that becomes quite confusing, especially for clinicians, because it's difficult to communicate to patients, and it's also quite confusing that you may find low-risk individuals that still develop the disease and high-risk individuals that don't develop the disease. So still quite confusing. But again, it's a new tool that we now have that we didn't have before, and that it helps us have a much more tailored, specific approach for treating patients and diagnosing patients. So how does this work in practice? And this is more to do with the second part of this workshop. So usually the first thing that we do, and I'm going to sort of skip through this. I'm not going to go too much into the details because it will be a lot more detailed in the sheet which I have put in the learning resources for this week. So if you go to the learning resources of this week, you will see this presentation, and you will also see a PDF document with the practical that we're going to do after I stop talking. So the idea is that from the 3 or 4 million variants that we might have, we want to prioritize them according to their most likely chance of contributing to risk. So we can filter them according to OMIM, which is a very well understood, very well known database that classifies genes according to their likelihood to contributing to Mendelian disorders. So if you have a disruptive mutation in an OMIM gene, you are more likely to develop a disease. Okay? This is what it really means. And as I say, the point of genomics is to compare and match what we see in the patient with existing databases to see whether that specific variant actually has been identified before, and to produce a specific phenotype. That's really what we are doing. So we are matching what we find in the patient with what databases know about specific variants. The problem is that for most variants, we don't have information. So the vast majority of the variants that, as of today, we have, they're what we call variants of unknown significance, meaning we don't have scientific evidence as to what their potential functional effects could be. So that's very limiting. But we are hoping that that will change with time. So here, I'm just giving you an example of an oming entry for gene MGH2. And it has found that if you have this gene mutated, you can have colorectal cancer or all of these other syndromes. There's another database which we might look at called ClinVar, which again, it tells us and helps us identify whether a specific variant has been identified before as being pathogenic or not. And there's another database called NOMAD, which basically looks at the frequency at which a particular variant has been observed in other populations from the world. So NOMAD is composed by about 150,000 genomes from populations across the world, and is able to tell us whether a specific variant that we observe in our patient is frequent or infrequent. The scientific basis for us to understanding whether a variant is frequent or not is that infrequent variants are more likely to be pathogenic because there's a negative selective pressure against them, which makes people carriers of highly pathogenic variants to not pass on those variants into the following generations. They don't have children, for instance. So the idea is that highly pathogenic mutations tend to be low frequency. And these database allow us to identify the frequency of known variants. And this is very, very useful. So, yeah, I mean, basically, the cutoff for us to deciding on whether a variant is highly frequent or infrequent is usually 1%. So if the frequency of a specific variant in NOMAD is less than 1%, then we say that it's uncommon. And that's more likely to be pathogenic. And so we need to use a series of algorithms. I'm not going to go into the detail, but basically the idea is that you want to predict for an observed variant whether it's going to be pathogenic or non-pathogenic. And then you look at databases such as oming, ClinVar, and then you bioinformatically try to predict the effect, whether it's going to break the gene or it's going to lose the function of the gene, or it may cause a frame shift in the protein. You also want to look at the family inheritance, and there are bioinformatics algorithms that help us predict the effects of a specific variant. There's also another concept I want to bring forward. One of them is what we call primary finding. So whenever you analyze a patient, the genome of a patient, you are looking for, let's say, for cystic fibrosis. So what you do, you look at the cystic fibrosis gene, and then you try to see if there are any pathogenic variants. That would be the primary finding, whether the reason for which the genome sequencing was indicated is ascertained or not. But also, we can also find secondary findings or incidental findings. Because when you do the whole sequencing of the genome, you're going to basically look at every gene in the organism. You may find other variants, other mutations, other pathogenic variants that you weren't expecting. Or, typical secondary finding is that you find that your dad is not your real dad. which I think like 6% of the UK population don't have the that they think they do have. So it's quite, and that is something, that's an incidental finding, because it's something that you see in the genetics that you weren't expecting, and yet it has a massive impact in the patient. Okay, so so that's why I want to differentiate, because I mean another example is with MRI, right? So you basically try to do an MRI of your lungs because you want to see the pneumonia a patient has, and then you find a little lump there, potentially a tumor, right? So you weren't expecting, but then because you perform that technique, you find something you were not expecting that may have a strong impact. So that's a secondary finding, okay? And sometimes secondary findings are indicated, sometimes they are not. All right. So this issue of variants of unknown significance is still developing. As I said, these are variants for which there's not sufficient information to assign whether they are pathogenic or benign results. Are they worth reporting? Probably not, because there's so much data and information anyway that you get that it may add unnecessary stress to the patient. So it's usually left to the discretion of the clinician. Anyway, so to finalize this presentation, hopefully I've given you a very, very brief taste of what clinical genomics is like. You might not get that taste ever again, so hopefully that will be a positive taste for you. I've basically told you that genomics is about identifying differences in the genome of the patient and then comparing it against reference data to see whether those variants that we see are likely to be pathogenic or not. And because we have so much data for each patient, everything has to be done on the computer and looking at databases. So it's quite bioinformatics intensive. And we need to make sure that the bits that are reported that are relevant to the judgment of a clinician. In other words, there's so much data that a lot of the time less is more, so that we avoid over-reporting and adding unnecessary stress to the patient, because not all pathogenic variants are worth reporting, and there are many variants of unknown significance that will hinder our understanding of the patient's genome. Okay? Hopefully you have any questions. I'm happy to answer any questions. Any comments? Did you learn anything new today? More or less? What was your favorite bit apart from secondary findings?
I liked all of it.
Thank you. Polygenic inheritance. Good. Okay, so I'm going to stop the recording. So the recording is going to be available.